Enzyme structure. The objectives of this video are to learn how the structure of enzymes helps with their function of catalyzing chemical reactions, and to understand the induced fit model. So, how do enzymes perform their function of catalyzing reactions? It is actually through a site of the enzyme known as the active site. The active site is the functional region of the enzyme. It is the site where the substrate binds in order to undergo the reaction. The active site is made of a small number of amino acids, and it is a small depression within the enzyme. So the enzyme has a globular 3D shape, as you can see, and the active site is a small depression. It is like a cave on the surface of the enzyme. And this is where the substrate binds in order for the reaction to be catalyzed. So when the substrate binds to the enzyme at the active site, it fits inside of it to form what is known as the enzyme-substrate complex. When the enzyme-substrate complex is formed, it is stabilized by temporary bonds in order for the enzyme to perform its action on the substrate, and then the substrate leaves after the reaction is completed. And of course, the enzyme is unchanged here. Here, for example, we have the enzyme. This is the substrate. It fits into the active site. So this is the active site. In the active site, there are 6 amino acids that are forming bonds with the substrate. After the reaction is complete, these bonds will break and the substrate will actually be turned into the product. So the product is going to leave the active site of the enzyme, and the enzyme goes back to its normal shape. Models are what scientists try to make in order to explain how something happens. The induced fit model of an enzyme is a model that scientists have come to agree that is what happens in reality inside of the body. So scientists agree that the active site is not actually always there, but it forms as the enzyme and the substrate interact. Enzymes are flexible. When something happens, a change in their environment, they respond to it by changing their shape. So here the change in the environment is actually the substrate approaching. So as the substrate approaches the enzyme, this is a change in the enzyme's environment. The enzyme is going to detect this change and respond by changing its shape and forming the active site. And this is only possible because the enzyme is flexible. So the enzyme starts to fit around the substrate in its active site. That's why it's called induced fit, because the enzyme begins to fit the substrate in it when the substrate approaches. This is induced by the substrate approaching. And while this change in shape happens, the enzyme is putting strain on the substrate. You saw that the enzyme forms some bonds with the substrate. It puts some strain on the substrate to cause changes in it, and these changes will lower the activation energy to allow the substrate to turn into the products. This is actually what happens. This is the enzyme. The substrate approaches, so the enzyme is induced to make this active site. The substrate fits into the active site. This is the lock and key mechanism. They are like a lock and key. The substrate fits into the enzyme. The substrate is like a key and the active site is like the lock. Of course, the enzyme is going to change its shape in order to fit this. The enzyme puts strain on the substrate in order to lower its activation energy, and now the reaction can occur. The activation energy is lowered enough for the substrate to be able to cross it, and so the substrate turns into the products, and the products leave the active site of the enzyme, and the enzyme goes back to its normal shape. So it starts with enzyme plus substrate, then we have the enzyme-substrate complex, and then we have the enzyme plus the product. And throughout all of this, the enzyme remains unchanged.